Okay, we're going to start our section of chapter 7 on transport, and we're going to start with passive transport. So, passive transport is diffusion of any substance across a membrane with no energy investment. Okay, and this is why it's passive. If you're very passive, you have no energy, you're not exhibiting any energy. So it is always going to be without an energy requirement. So diffusion is the tendency for molecules to spread out evenly into the available space. Mother Nature always likes an equilibrium. We want to try to get to that point. And so nature is always going to push towards an even distribution of molecules. Again, molecules move by random molecular motion, but diffusion of a population of molecules can be directional depending on concentration. Okay. When they do reach equilibrium, it's not static, however, it's a dynamic equilibrium. And it just means equal molecules are crossing the membrane in one direction as in the other. So if we look at the diffusion of, here we've got some molecules of dye and water, and there is a high concentration on one side and a low concentration on the other. So the molecules are going to move towards the area of low concentration. We call this moving down the gradient. And it'll be explained more on the next slide. So the net diffusion is going to be this way until there's an equal distribution of molecules on both sides. So this is my membrane, right here, membrane. Till there's an equal distribution of molecules on both sides. But again, it's not static, it's dynamic. Okay, the molecules still move, but you have no net movement in either direction. If you have two solutes, diffusion can occur in both ways. So diffusion of the orange solute would be in this direction, down its concentration gradient. The purple would be moving this way across the membrane. And again, they spread out evenly until we reach a dynamic equilibrium of both particles. So as I just mentioned, substances diffuse, i.e. no energy, down their concentration gradients. Okay, so this means from an area of high concentration, the brackets mean concentration, toward an area of lower concentration. Okay. Again, no work is done via diffusion to move the substances down their concentration gradient. Okay, and so therefore diffusion is always passive because it does not require energy. A special type of diffusion is osmosis. Okay, osmosis is the diffusion of water, and it just refers to the diffusion of water. Okay, remember all those, all those times your mom said, oh, put the chemistry biology book against your head and you can learn by osmosis. No, that would be learning by diffusion. Okay, the only thing that moves via osmosis is water. Okay, so again, and because it's passive, because it's diffusion, it's passive. So water is diffusing across a membrane down its concentration gradient, but it's also equalizing the concentration of solute. So it moves from a region of lower solute concentration, which means higher water, to an area of higher solute concentration, which means lower water. Okay, until the solute concentration is equal on both sides. So if I have low solute, that means high amount of water. Okay, and that goes towards high solute, which is a lower amount of water. Okay, so again, we're going down the concentration gradient, high to low with the water. This is just a little picture of, so we've got a membrane here in our U-tube. Look, it's U-tube. And you could see there's a higher number of solute molecules on this side, so the water is going to have a net movement in this direction until there's an equal concentration of solute molecules. So the solute molecules themselves can't diffuse across the membrane. Only the water molecules are small enough to move via osmosis across the membrane. And so eventually you'll get an even distribution concentration based on volume. So let's talk more about osmosis and water balance in cells. Tonicity is the ability of a surrounding solution to cause a cell to gain or lose water. And this is very, very important in biological systems. And this is actually what our first lab is going to be about. So there are 3 types of tonicity. There's an isotonic solution, which means the solute concentration is the same as that is inside the cell. So there is no net water movement across the plasma membrane. If you have a hypertonic solution, the solute concentration is greater in the solution than it is in the sides of the cell, and the cell will lose water. Water will go out of the cell. A hypotonic solution, the solute concentration is less than that inside the cell, and the cell will gain water. Okay, so what does that mean? Let's look on the next slide. So the easiest one to look at is the isotonic. Isotonic means equal. Okay, so again, it's not that there's no net movement. We're at equilibrium, but it's at a dynamic equilibrium. And so the surrounding solution has the same concentration as inside the cell. And so water moves in and out. Okay, in and out. Here's our animal and our plant. But there's overall no net movement. Okay. This is a normal situation for a plant, for an animal cell, but it's actually not a normal situation for a plant cell. So this is normal for a plant cell. So let's move over to hypotonic. Okay, so hypotonic means that the solute concentration in the solution is less. So there's more water in the solution. So water is going to move into the cell. By the way, these are red blood cells. And the cell will swell until it bursts or lyses. Okay. And an easy way to remember hypotonic solution is hypotonic solution, the water moves in and the cell gets fat like a hippo. Okay, hypotonic, like a hippo. Okay, and then this solution where the water moves into the cell in a hypotonic solution, this is normal. We call this turgid, normal for a plant cell. We have the cell membrane pressing against the cell wall and keeping the plant. And this, when your plant is fully watered and happy to go, it has turgid cells. Okay. The reverse of this is a hypertonic solution. And this is when the solute concentration is greater outside the cell, so that water moves out of the cell into the solution. This causes the cell is losing water, so it shrivels up or shrinks. Okay. That's in an animal cell. In a plant cell, we call this plasmolysis. And you see the cell membrane is pulling away from the cell wall, which is not a good situation at all for a plant cell. Okay. And an easy and not remotely politically correct way to remember hypertonic is if you have a hyper kid, you take them to see a shrink or a psychologist. So the cell shrinks if it's in a hypertonic solution. Okay, hypotonic, fat like hippo. Hypertonic, shrinks. So again, hypertonic and hypotonic environments can create problems for unicellular organisms like amoebas and parameciums. Okay, and so they have to find ways to regulate this, and through osmoregulation, control of solute concentration and water balances. And this is an adaptation for most protists who live in an aqueous environment. They're unicellular, they live in water, but they have a very high concentration of solutes inside of them, okay? And the paramecium is hypertonic to its environment, which means it's in a hypotonic solution. So again, if it's hypotonic, the cell, our poor little paramecium, is going to get fat like a hippo. So it keep taking on water. So it's evolved a method of getting this water out, and that is via a contractile vacuole. And what happens is, let's show this contractile vacuole, there it is. It acts as a pump. So it collects water, it's a vacuole, it will collect all the water from the cell, and then it shoots it out of the cell. And then that causes the cell to shrink back to its normal size. It doesn't get all swollen. Of course, it's a continual process because because it's in a hypotonic solution, it'll still always be taking on water from its environment. Okay. Cells that have, so plant cells, again, we talked a little bit about this already in the previous slides, but again, hypotonic solution is what we want for our plant cells because it gives them a turgid or a firm consistency. The plasma membrane is pushing against the cell walls because there's plenty of water inside. And this is how plants manage to stay upright. Again, if you have the plasma membrane not pressing hard up against the wall, the plant will become flaccid or limp. This is an isotonic again. And that will cause the plant to wilt. If you wilt, you need more water. The water goes into the cell, you get turgid, and you're all set. Okay? If you put the cell in a hypertonic environment, as we said before, the plant cells lose water and the membrane pulls away, and this is called plasmolysis, and that usually leads to the death of that cell. All right, so all diffusion is passive. All diffusion is passive. But sometimes things need help to cross the plasma membrane, even if it's going with its concentration gradient. And so passive transport can be aided by proteins embedded in the membrane, and we call this facilitated diffusion. So there are proteins that transport molecules across the plasma membrane. Okay, there are two types. The first type is called a channel protein. And a channel protein basically makes a little corridor that allows molecules to cross the membrane. So normally molecules that can't cross the membrane, things that are polar, ions, they can't cross the membrane because the interior of the membrane is nonpolar. So what these channel proteins do is they provide a little tunnel through the membrane so that polar and charged molecules can get across, because there are lots of polar things that need to cross the plasma membrane, not the least of which is water. So there are special channel proteins called aquaporins that allow water in and out of the cell. Ion channels allow ions to cross the channel. These are very involved in nerve impulses, as we'll see in a later chapter. And then the second type of facilitated diffusion is carried out by carrier proteins. And carrier proteins are much more specific. They allow one specific substrate to cross the membrane. And it does so with a subtle change in shape that moves the molecule across the membrane. If we look here, a channel protein basically just has a tube through the center. And basically, if you fit, you can go through. It's not discriminatory whatsoever. Carrier proteins are a little bit more specific. They bind to a specific solute Okay, solute goes into the binding site. The binding causes a change in structure, and that will result in a change in function, obviously. And then the solute is released to the other side of the cell. Okay, so that's it for passive transport. And we're going to move on in our next screencast to active transport. So get psyched for that.